Hey everyone, how you all doing this week and this weekend so far? I hope you guys have been all doing good, and I hope studying has been going well as well. I know it's been busy, right? It's been a busy couple weeks, so hopefully we'll get through this together, and you all do well on the next exam. Um, and yeah, best of luck on studying and everything that you would do in this class. So for today, we're going to talk about chapter 7, which is talking about the membrane of the cell. So the membrane of the cell is like the boundary of the cell. Okay, it separates the cell from the outside environment. It's very crucial to the cell for its survival and for its maintenance as well. So we're going to talk about the anatomy of that membrane, what is it made out of, and the different functions that this membrane does. Okay, so let's get started. So the membrane is separating all the cells from their external environment and surroundings. Okay. Um, they do exhibit something called selective permeability, which means not everything can cross inside the cell. Okay, so think about the membrane, or we call it the plasma membrane, right? It's like a gate. Okay, and whenever you have a gate that's surrounding, let's say, a city, would you want anybody to come into that city, or would you want to check who's coming in and out? And maybe only allow friends or reject enemies. So this is what we mean by selective permeability, right? So it's allowing things to come in and some other things not to come in. um, so there are several ways that the membrane allows things to come in and out. Some things can go in and out without any gates, so they can just uh, like just flow in to the cell, depending on what they're made out of. Okay, so remember, the plasma membrane of the cell is made out of what? So it's made out of a what? Dual layers of what? Phospholipid bilayer, right? So it's got basically the head and the tail, right, on each layer. And where is the head facing? The heads of the first layer is facing the external environment, while the tails are facing the inside. Okay. On the other layer, you also have the head also facing the inside of the cell, and the tail facing the other tails. Okay, of the other um, layer of phospholipid. Okay, you will see a depiction of that in the next slide. Okay, so that's basically how some molecules pass through these two layers, because these two layers have a hydrophilic side on the external side of the cell and a hydrophobic on the middle part of them. Okay. Um, Cells also sometimes require proteins, what we call them transport proteins. So think about them as like gates. Okay, so those transport protein are gonna allow some things in, some and they're gonna basically um, facilitate the passage of those molecules inside the cell. Okay. So we're going to ask, how does the plasma membrane actually regulate the inbound and outbound traffic, right? So there are like lots of things going on outside of that plasma membrane on the external environment, right? So the cell actually uses, the something called passive transport. Passive means it's passive, so it's not requiring any type of something active or something that's requiring energy. Okay, so it's like going with the flow, if you will. Okay, So there's no input of energy when we say passive transport. There's no input of energy. Okay. And later on you will learn when we say active transport. Okay. Active. Just put it in your head. Active means we need energy to allow a certain molecule to go inside the cell or outside the cell. Okay. And this is achieved through what mainly? As you know by now, what's the energy molecule of a cell? It's ATP, right? So whenever we want to bring in or out, ATP is being used as the energy molecule. Um, Usually what's happening is ATP is being hydrolyzed, right? So the cell is basically using that ATP and splitting it into the ADP form, right? The adenosine diphosphate, right? Instead of triphosphate. So there's always that phosphate being released, okay? If you remember all of that from the previous chapters. Okay, um, so again, some small molecules use that active transport I was talking about, right? So they need that energy or ATP, right, to be able to transport that protein, okay? And while other larger molecules, so when we say larger molecules, we're talking about like amino acids, we're talking about carbohydrates, things like that. Basically, they basically move in and out of the cell um, using what we call um, exocytosis or endocytosis. So that's basically, do you remember from last chapter when we talked about the phagocytosis is when the cell basically like grabs onto the whole whatever it's trying to digest and like basically engulfs the whole thing inside. So that's a form of what endocytosis, okay? When we talk about exocytosis, it's from the word exit, or we're trying to bring something outside of the cell, okay? Endo, inside the cell, okay? So we'll talk about all of those in detail in the next few slides. So here we go. So we're going to talk about the anatomy a little bit of that phospholipid bilayer. So as I mentioned to you, right, we've got this hydrophilic head, right, and what is it gonna basically like, react with? Polar molecules, right? Well, we have the hydrophobic tails, right, facing each other right here, okay? And this is what they look like when we look at them, as far as their molecule shapes. Do you remember that from previous chapters? Okay, so these are the two tails, right, facing each other, and then you have the polar side or the hydrophilic side facing the outside. Okay, so we call this amphipathic molecules, okay, because they're containing basically both type of um, ability, which is hydrophobic and hydrophilic, right? Hydrophobic meaning water-fearing, or kind of like does not mix with water, and hydrophilic is water-loving, or it mixes with water. Okay. So if I ask you like what are fats, right? Fats are what? Are they hydrophilic? Can fats mix with water? No, right? So they would be hydrophobic. Okay. So the hydrophobic tails basically of those phospholipids are inside, as you can see, the membrane. Okay, and those tails are basically going to allow some non-polar molecules. Remember this point, to come in the cell without using any type of gate or protein channels. Okay, so remember this point. Okay. All right. So this looks very busy, obviously, right? There's a lot of things going on here. But don't worry about it because we're gonna explain each one what that is, like little by little. But just to give you a little summary of what's going on here. So this is a cell, right? And it's showing you the different type of transportation that the cell can use to transport things in and out. So we talked about the passive transport, which uses what? No energy, right? No ATP. We talked about the fact that there are transport protein acting as gates, right, allowing things inside, and then there is also some molecules that are being used during the active transport. We will talk about, when does this happen and why does this happen. Um, this is using the ATP as energy, as you can see here. And then this is showing you also the endocytosis, which is basically allowing things in, so like big molecules, using vesicles, right? We talked about this last chapter, I believe. And then basically exocytosis is when we also want to basically form some type of proteins or some molecules, and we want to basically get them out of the cell, okay, after they're synthesized, let's say in the endoplasmic reticulum, okay? So this is done by what? Exit or exocytosis, is that right? Okay. So hopefully this is making sense a little bit, just to give you a brief summary of everything. Okay, so let's see here. We're gonna talk about the membrane, the membrane model being a mosaic of lipids protein model. Okay, so let's see here. So lipids and proteins basically are the main components of the membrane. Okay, we can see that here. So these channels right here, do you see that? These are all made out of what? Proteins, right? So you got integral protein, which is integrated inside that membrane, plasma membrane. You have some peripheral proteins here. You have, some like cholesterol. Do you see that? The cholesterol's in between. Oh, sorry about that. Um, these are embedded in where? They're gonna be on the hydrophobic, right? So cholesterol is made out of fat, right? You have some glycolipids, okay, on the outside, as you can see. So glycolipids, we will talk about them in a bit. So they're made out of what? Both carbohydrate and fat, right? So from the name you can tell a lot of things. So glyco, kind of like glycogen, right? And lipid, okay? So that's how we form this. Now you also have glycoproteins. That's formed out of what? Also carbohydrate, which is coming from the word glyco, okay? And protein, okay? So um, let's see here. So phospholipids in general, right? As far as the membrane goes, we call them amphipathic, right? Yeah, amphipathic. Sorry. Basically they contain a hydrophobic and a hydrophilic um, side, right? We talked about this already. Okay, so proteins are basically kind of not randomly distributed in the membrane because they have certain location that they need to be in. Okay, but they do, just so you know, they do move. Okay, so they're not like just immobile. So think about the membrane as how can I think about it? A little bit like a jelly kind of thing. Okay, so it's like kind of mobile all the time and kind of fluid-like structure to it, okay? And so the things that are on it, such as like proteins, protein channels, and other molecules that are like integrated inside that membrane, they're being like, shifting around and moving around such as, whatever the cell wants them basically to do and where it wants them to be, okay? All right, so let's see here. So we talked about all of these. I know there's a lot of repetitions on those slides a little bit, but just to repeat those ideas and basically so you guys can kind of study that. Um, let's see here. Okay, so I wanted to tell you that those membranes are being held together by basically weak hydrophobic interactions. Okay, they need to be weak because that membrane, remember, it needs to be fluid, so it can't be like immobile. Where if you have like a very strong hydrophobic interactions, then it's not gonna be able to move as freely. Okay, So as I mentioned, those proteins, right, and those lipids that are located on the outside of the molecule, they move sideways, right, within the membranes. Again, so this is basically a repetition of what I said. Okay. So this is just showing you like an experiment. I think this is also in the book. We put it here just to show you and talk about it a little bit. So they made an experiment here, some scientists, and they basically mixed some mouse cell and human cell together. And the purple on the outside and the pink on the outside are just like kind of like what we just saw on the membrane. So they're like protein channels and glycolipids, glycoproteins, and so on. So when they mixed them and they made a hybrid cell, those mixed proteins actually, like you see how they shifted around. Do you see that? Now you have like a purple, then a pink, then a purple. So they do shift around. You see that? They don't stay immobile. Okay, they don't. Okay, so we're gonna talk about why the membrane is actually like fluid, and how does it actually sometimes solidify just a bit, right? It's not good for it to solidify too much. So just so you know that as the temperature, right, cools, so as temperature basically becomes colder, the membrane switches from a fluid state to a solid state. Okay, so that's just the way it is, and what you're seeing here is basically like the unsaturated versus saturated tails. So remember from our previous chapters, right? The unsaturated fats, they have what do they have? Do they have single bonds or double bonds in between? They have the double bonds, right? And when they form the double bonds, they're basically creating those kinks, right, in the molecule. And so those tails, you see how they have like a kink in one of their legs, and that's um, because of the unsaturated tails um, have double bonds, right, and they're basically kinking, and when that happens, right, um, we have them as fluid instead of solid at a certain temperature. Okay, so think about this as like olive oil, right? Olive oil is made out of what? Unsaturated fat. And so is olive oil solid at room temperature, or is it fluid? It's fluid, right? How about like butter or animal fat? It's solid, right? So this is the case of saturated fats. So they pack tightly together because there's no kinks. Okay, that's why we call them saturated fats. Okay. So let's see here. But let's see. So the temperature, just so you know, at which a membrane solidifies, okay, it really depends on the type of lipids. So if you have more of saturated fats, it's going to be different than having unsaturated fat where the membrane is going to stay more wet in the liquid form. Okay. So again, if a membrane is rich in unsaturated fatty acids, such as this model here, it's going to be found as what? More fluid. Okay. All right. So hopefully this is making sense a bit. I know it's new information, so let's continue. So we talked about a few slides ago that cholesterol is embedded next to those tails, right? So the cholesterol's job is basically it reduces the membrane fluidity at moderate temperatures. So think about cholesterol as like a buffer, okay? Buffer type of fat. So it's really like controlling the amount of fluidity that the cell has at a certain temperature, okay? So it really like reduces the fluidity at moderate temperatures, right? But actually also hinders the solidification at low temperatures by disrupting packing as well. So you can see that it's acting on both, fluidity and solidity, okay? So we talked about how membranes must be fluid, right, to work properly. Okay. So the fluidity obviously does affect, how things are flowing in and out, and the movement of transport proteins across that membrane. Okay. Um, so if they're too fluid, then they cannot support the protein function, and that's why we have, like, the different, like, cholesterol, right, as a controller. That's why we have, the saturated fats, fatty acids, right? Those are gonna make it like not very fluid. Okay, so it's kind of like everything is trying to equalize itself for that membrane to be very stable. Okay, so organisms in extreme temperatures, right, have adaptive differences in membrane lipid composition. So those organisms have, like, adapted to live in those tough conditions, right? So some regular organisms that go in at a certain extreme environment might not be able to withstand, like, the different temperatures in the environment and so on. Okay. So think about the membrane as basically a tile mosaic. So this is a model of the tile mosaic. So it's pretty cool, right? So it is a collage of different proteins, right? Often clustered in groups. Okay. So looking at that membrane, you will find, like, tons of just uh, you know proteins embedded in it, and glycoproteins, glycolipids, carbohydrates. You know, a lot of gates and channels, and all of these things, which is amazingly, they do a specific job, and they work all together for the ultimate benefit of the cell. Okay. So think about the phospholipids as the main fabric. Okay, so it's the main cloth of the membrane. Okay, but those proteins that are basically embedded in the cell, and the membrane, I mean, they're the ones that are doing the functions. So they're basically the soldiers, if you will. Okay, that are, like, basically allowing things in out, and workers. Okay. Okay. So this is also just talking about basically the type of proteins, right? We have peripheral proteins. Peripheral, sorry, means what? They're basically on the side of the membrane. Okay. Do you remember that from the first slide? I don't. What you're seeing here is an integral protein. Why is it integral? Because it's embedded. So it's being integrated inside the membrane. Do you see that? Okay. So the peripheral are basically on the surface of the membrane, whether on the outside or the inside, and it's not being shown here. The integral is basically penetrating the hydrophobic core. Okay. So we call these basically transmembrane proteins. So they're being basically, they go through the membrane, so they're transmembranous proteins. Okay. The hydrophobic regions of an integral protein, basically, they have, obviously, what? So when we say hydrophobic regions, we're talking about what? Non-polar, right? Non-polar amino acids. Okay, so they're found in, basically, they're coiled into alpha helices, as you can see here. Okay. All right, so we're going to talk about the different functions that happen in a membrane. Okay, so you have transport, enzymatic activity, signal transduction, cell-cell recognition, intercellular joining and attachment to the cytoskeletal and extracellular matrix. Okay, so this is just a little brief of each one. We're gonna talk about, like, each one's little by little, so you can kind of understand what they are. Okay, so the first thing is basically the transport. Okay, let's see here. Okay, so let me just give you a little brief here. before we move on to the next stuff. So transport basically, it's what it says, right? It's when the proteins are transporting molecules in or out, whether in a passive way, passive transport, or active transport, which is what you're seeing here using that ATP. Okay. They can also those membrane proteins can function as enzymes, right? So when those enzymes detect some certain molecules, as you can see here, then they produce changes inside that membrane, depending on whatever whatever the molecule is, right? So you can have basically like let's say insulin, right, from that's being produced from the I guess I forgot where insulin is produced, but it's usually like in the either the liver or the adrenal gland. I believe the liver. But whenever that's produced and that's being detected by those enzymes, then you have some changes going on in the cell, right? Then you have also signaling molecules. So that's also like you can also think about insulin like that too, right? So when that signal molecule is detected by that receptor protein, it does a transduction signaling inside the cell. Usually, like it could even like communicate with the nucleus of the cell, okay, through different like molecular cascades, okay, cascades. I mean, during also cell recognition, right? So there are two, nearing by cells. One will recognize the other using those, proteins. Like for example, they can use their glycoprotein. So it's kind of like a puzzle, and they can recognize their shapes based on that. Okay, you have intercellular joining too. So sometimes if they want to like join together tightly, like when we talked about in gap junction and tight junctions, cells use those proteins for that too. Okay, and then you have also the ability to attach to cytoskeleton. Remember those things we talked about too. Like the microfilaments and microtubules and things like that. Okay. Okay, so I'm gonna talk about this for a sec. This is actually in the book. So this is talking about how the cell surface proteins are important in the medical field. So we're talking about HIV patients, right? So basically, the researchers and scientists have discovered that the cells usually have actually the immune cells of patients and just the human population in general. They have a protein, uh, cell surface protein called CD4. Okay, and you will probably learn about this more when we talk about like immune system and immunology. Okay, so they also found that HIV not only binds to the CD4 protein, but it also binds to something else called CCR5, a cell surface protein, and that's actually a co-receptor. Okay, so in order for the HIV to enter the cell, the immune cell, and hijack it, basically, it needs to basically binds to both CD4 and CCR5. So if it cannot bind to one of them, then it cannot enter the cell and hijack it, and thus you don't have the damage that HIV does, which leads to AIDS, right? So what the researchers discovered is that hey, if we attack the CCR5, if we block that receptor, which is called CCR5 on immune cells, human immune cells, okay, we can basically block HIV from hijacking the cell, okay? Um, but you might think like, why not CD4, for example? Why CCR5? Because CD4 is basically extremely important for the immune function of, of like all the immune cells. You need CD4, otherwise, your body can't fight viruses and bacteria, okay? So the fact that they were able to, you know, um, find out like this about CCR5, this allowed them to basically develop drugs which allowed them to mask the CCR5 protein and thus blocking the HIV from entering those cells. Okay. Oh, and this is here we go. So this is what we're talking about with this. So you can see the CD4 right here and CCR5 here. You can see that HIV virus. You see that? Okay. So it needs both to enter the cell. This is the cell right here. Okay. So if you have a CD4 but no CCR5, that HIV cannot enter. Okay. All right, so let's talk about the role of membrane carbohydrates in cell recognition, right? So these cells can recognize each other, using these glycoproteins, okay? Um, so they're kind of like a, a match to fit, okay? Kind of like a puzzle. And that's how they recognize each other, okay? So membrane carbohydrates, I mentioned this before, right? If they're bound to lipids, then on the surface of the membrane, then we call these what? Glycolipids. If they're bound to protein, we call them what? Glycoproteins. Okay. Now carbs on the extracellular side of plasma membrane, right? They do vary among individuals, right? And obviously the cell types. So not all the cells in the body function the same way, and the location and the functionality will have differences on those molecules. Okay. All right, so let's talk about basically the distribution of those molecules. So what you're seeing here is the ER, endoplasmic reticulum. You see the Golgi apparatus, right? That's the UPS or the transportation company. Okay. We see that those molecules, the glycoproteins or glycolipids, are being manufactured by the ER, endoplasmic reticulum, right? And then they go through the Golgi apparatus, and then eventually, basically, uh, uh, they get formed, and the vesicle will carry those molecules to the outside of the cell, and they are basically or they get excreted using what? Exocytosis, right? We talked about this in a bit a while ago. Okay. After this happens, those molecules basically get embedded on the outside of the membrane. Do you see that? Okay. Hopefully this is making sense now. So let's see here. So the plasma membrane is basically selectively permeable, as we talked about. So it's regulating all that traffic in and out, and it's choosing specifically what it wants to get in and out. Okay, using those gated channels, proteins, and the different membranes or workers, if you will, around it. That's helping it, control things in and out. Okay. So the hydrophobic is the nonpolar, right? It's gonna allow what kind of molecules in? Nonpolar, right? Hopefully you know this by now. So these will basically be able to dissolve in the lipid bilayer and pass through the membrane rapidly. Okay. Some examples would be what? Hydrocarbons, right? Do you remember that? So all those CH, carbon hydrogens, chains that we talked about, the rings and all of that. So these are called hydrocarbons, and these are nonpolars. They're gonna be able to pass through that lipid bilayer easily, rapidly, okay? CO2 is the same way. O2 is the same way, okay? And guess what? Do we need to actually have O2 and CO2 going in and out fast? Yes, we do, right? That's what we breathe. When you breathe in, you're getting that oxygen in, and it's being distributed to the whole cells in like less than a second. CO2 the same way. When you're trying to breathe out, you need to basically the cells need to get out all that CO2 and throw it out, right? So that's why it's important that they pass through fast. Now the hydrophobic right interior of the membrane impedes the passage of hydrophilic, right? Nonpolar is not going to allow the polar stuff in easily, okay? Guess what? This is where you have those protein channels coming into play. So we've got sugars, water, and ions that pass slowly, okay? Um, they're gonna be able to pass through the passive transport or the active transport. So these are the two ways that that things go in and out, okay? So transport proteins basically, again, they allow those hydrophilic substances across the membrane, right? Like we just talked about. So like sugars, like water, ions, right? All of these things, okay? Let's see here. So the membrane has channel proteins, so they're called channel proteins. When we talk about channel proteins, we're talking about what? It's a channel. Okay, so it's like a little pathway, direct pathway that's allowing things in and out. But however, just know that these channels are specific for things. Okay, they're not gonna allow certain things that are not recognized in that passage or channel. Okay. So although it's a little pathway, right? It's still not allowing things that's not recognized. So the ones for water specifically, they're called aquaporins. Okay, so remember that aquaporins, agua. Okay, so agua for water, aquaporins are the channel proteins that are allowing the water inside the cell and outside. Okay. Now another type of or proteins that transport things. They're called carrier proteins. So as it sounds, those proteins, they carry the molecule. Okay, then what they do is they change their shape. Okay, and then what they do is they after they change their shape, they put that molecule inside the cell. Okay, same way if they want to throw something outside, they carry it, then they change shape, and then they throw it outside. Okay? Okay. So again, these transport proteins are moving things slowly. They're not rapidly like the non-polar hydrophobic stuff, such as CO2, O2, hydrocarbons. Okay? So these hydrophilic molecules, right, such as glucose, they're carrying, like in this case, if it's glucose carrier protein, right, they're transporting only glucose, right? So they're specific. Now, if you try to give them fructose, although I don't know if you remember this, but fructose and glucose are very similar, except the fact that fructose is an isomer of glucose. Do you remember what isomers are? So they're structurally different uh, than glucose. Okay, they still make up the same uh, you know same molecules. So same amount of carbon, same amount of hydrogens, so on, right? But they're just their shape is different. So that transport protein is not going to allow fructose in, but it's going to allow glucose in. Okay, in this situation. Okay. All right, so we're gonna talk about diffusion now a little bit. Okay, so that's a way of the cell to diffuse things. So it is the tendency for molecules, okay, to spread out evenly into the available space. So you have basically a how can I explain this a little bit? You have let's see here. Let's think about it like some bottle filled with water, right? And you're trying to dump the water outside it, right, into the space. And so what is going to happen is, that water is going to continue to drop out of this bottle as long as you have it like basically tipped over, right? So diffusion is just moving those water molecules out, okay? So just know that at dynamic equilibrium, okay, what's happening is that the molecules that are crossing the membrane in one direction is equal to, right? That's why we call it equilibrium. So just the science is always about like names, and they all kind of like make sense when you think about them this way. Uh, so equilibrium means like, we have the things that are going outside is equal to the things that are coming inside. Okay. Okay, so this looks like a lot, right? But it's not that difficult. So we're gonna talk about diffusion a little bit here. So again, look at those circles, little circles. So these are basically some molecules, or you can even call them solutes. Okay, so think about them like, for example, just salt, or sodium chloride. Okay, so the molecules are, and this is a depiction of the membrane. Okay, and it's dashed, just so you know. This is a representation of selective permeability. Okay, so the molecules are trying to cross to this side. Okay, and so the arrow is showing you, going to the right. Okay, so this is the amount, the way the molecules are trying to diffuse. Okay, now when they start diffusing like a lot this way, and remember the membrane is dashed open, so these molecules can go in and out, right? When that movement is kind of equal, then this is when we have equilibrium. Okay. So you can also have like uh, obviously two different molecules, or three different, or even 1,000 different molecules are going in and out, right? And you can have that equilibrium as well. Okay, even though you have different molecules. Okay, you guys can watch all of these animations on your own. We're not gonna take the time right now for them. Okay, so let's see here. So passive transport, right, is diffusion of a substance across membrane with no energy. Do you remember that? Passive, no energy. Active, yes energy. Okay. So whenever we're talking about the passive transport, we're talking about the fact that diffusion of molecules are happening down their concentration gradient. Okay? So what this means is that the region they're moving into, okay, has a less amount of them, so therefore they want to move into that space. Okay? Less amount of density of that chemical. Okay? Again, there's no work during that passive transport. No energy that's needed. Okay? So just so you know, the rate of that diffusion, it just depends on the membrane permeability of that specific substance. So it's not really dependent on the amount of energy. Okay? So just, um, if a molecule is, for example, bigger, then it might require more, uh, you know, like more time for it to diffuse. Okay, just think about it this way for now. Okay, so we're gonna talk about osmosis. So just think about this and remember it. Osmosis. Whenever you're talking about osmosis, we're talking about the movement of water specifically. We're not talking about anything else but water. Okay, so it is the diffusion of free water. Okay, across a selectively permeable membrane, such as the membrane that's surrounding the cell. Okay, this is a little depiction of what's happening. So this dashed line is again that selectively permeable membrane. Okay, so what's happening here is you have water in that U-shaped tube, and you have some sugar molecules represented here by the green circles. Okay, so if you have a higher concentration of solute here, okay, what is the water going to do? The water is moving this way, okay? What's happening is wherever there is glucose molecules, okay, uh, the water molecules, which is made out of what? H2Os, right? So what they do, the H2Os actually get clustered, okay, around those glucose molecules, as you can see here, okay? And so what's happening is that the free water molecules can diffuse across the membrane from the region of lower solute concentration, okay, lower to higher, okay? So in this situation here, we have what? We have, it looks like we have glucose more on this side. Do you see that? Okay? So we have more water molecules on this side now. Okay? So where do you think the water is going to move? You have more glucose molecules on the right side. And we said that water follows the solutes. So if you have more higher concentration of solutes here, the water is going to move to the right more, okay? So the water is going to keep moving, just so you know, until the solute concentration is equal on both sides. That's called, again, what? Equilibrium, okay? So this is the movement of water, okay? We're not talking about movement of solutes here, but movement of water, and that's called osmosis. And there's an animation here. You can see that on your own. Okay, so let's see here. So we're going to talk about water balance of cells, which is very important, right? The water inside the cell versus the water outside the cell or in its outside environment. And we're talking about cells without cell walls. So which cells are we talking about? Animal cells, okay? Are we talking about plant cells? No. Plant cells have cell walls. Talking about prokaryotes? No. Okay? So tonicity, when we say the word tonicity, we're talking about the ability of a surrounding solution, right, to cause a cell to gain or lose water. So now we're talking about the effect of solutes on water, okay? Specifically on the surrounding solution. So surrounding the cell, the outside of the cell. So if a solution has a higher concentration of these solutes, okay, than the inside of the cell, what do you think water is going to do? We just talked about this in osmosis. So if you have more higher concentration on the outside, water is going to do what? It's going to leave the cell to the outside. It's going to follow those solutes. Okay? And if it's the other way around, if you have more solutes, like less solutes outside the cell, water is going to rush inside the cell. Okay? So again, tonicity of a solution depends on its concentration of solutes. We're talking about solutes here. Okay, so there are three types of tonicity of a cell. So we have isotonic solution, okay, meaning that the solute concentration, okay, of that environment is the same on the outside as the inside. So if you have the same, then water is not going to move. There's no net water movement across the plasma membrane. So another word would be there's no osmosis. Okay? So water is being diffused in both the directions. It's not no osmosis. I mean, you still have water movement, but it's like, equally. Okay? Just so we're not confused on this. Water is always moving in the cell. It's not going to stop moving. Okay? So in hypertonic solution, okay, so the solute concentration is actually greater than that of the inside of the cell. That's why it's called a hyper. Hyper is more, okay? So what happened during hypertonic solution? If there's more solutes, or let's say, I'm just going to give you an example, salt. If there's more salt in the outside of the cell, water is going to move outside the cell and follow that, where the salts are, okay? Now, cells without cell walls, such as animal cells, will lose water. They will shrivel, right? They will shrink because the water is coming outside of them, and they will likely die in hypertonic solution. Okay. Now, in hypotonic solution, it's the complete opposite, right? So what's happening here is that the solute concentration is less than that of the inside of the cell. So what is water gonna do? That's gonna rush inside the cell, because inside of the cell has also solutes, right? So basically what's happening is the cells, the water goes inside the cells, and then the cells swell and sometimes can lyse and burst, okay, in a hypotonic solution. Okay, so if they have cell walls, and we're gonna see like plants, for example, it will be a different story. Cell walls basically allow it's some type of a defense of the cell, okay, so it's not gonna allow the cell to burst, okay? And this is showing you what we just talked about, okay? I'm not going to repeat this because we just talked about it. Just look at it on your own, okay? Let's see, hypertonic or hypotonic environments create osmotic problems, right? For organisms that have cells without rigid, we just talked about this, right? Those rigid cell walls will allow the cells to basically withstand the water pressure, okay, that's being exerted on the membrane, okay? So osmoregulation, the definition of that is the control of solute, right, concentrations and water balance, okay? It is a necessary adaptation for life in such environments. So what you're seeing here is actually an example of paramecium. It's a unicellular eukaryote, okay? It's hypertonic to its pond water environment, okay? So it's got this little vacuole here that's contractile. So contractile means it contracts. So this actually acts as a pump, and it pumps the water out, okay? All right. You can watch that. All right, so we're going to talk about now the water balance of cell with cell walls. So these cells would be what? Cells of plants, right? Mainly. Prokaryotes, fungi, and some protists also have cell walls, okay? So these cell walls are going to help the cells maintain water balance, obviously, because they're tough, they're rigid, okay? So a plant cell in a hypotonic solution, what do you think is it going to do well or bad? It's going to do well, actually, right, in a hypotonic solution, okay? So it swells until the wall basically opposes uptake. That's what's happening, right? So the cell wall is going to oppose the uptake, and so we call this cell is now, I think you pronounce this turgid or turgid, I'm not really sure, but this means it's very firm, and this is the healthy state of most plant cells. When you give cells, plants water, sometimes they, you see them, they're dying and whatever. When you give them water, they become alive again, okay? It's because, that outside environment, right, and which is like around the roots, is basically, what? Hypotonic, right? It's water, it doesn't have solutes. So what's gonna happen is gonna, what? Rush inside the cells, okay? So if a plant cell and surrounding are isotonic, okay, which means isotonic, which means there's no net movement of water, right? Then the cell becomes flaccid, which is limp, okay? That's why it needs to be in this hypotonic environment. Okay, so in a hypertonic environment, right? That's the opposite of hypotonic. So the plant is gonna plant cells are gonna lose water. Okay, like always, hypertonic. It's gonna basically allow the cells to shrink. Okay, the membrane pulls away from the cell wall, right? In this situation. So remember, you have a cell wall in the plant, but under that cell wall you have a cell membrane still. It's not like the cell membrane goes away. It's still there. It's attached to the cell wall normally. But in a hypertonic environment, those membrane pulls away from the cell wall and create basically, you know, some basically some bubbles, right, around it, and and so this is a lethal effect of on the plant, okay, on the plant cell, and this is called plasmolysis. Okay, whenever you see the word lysis at the end, means something is dying. Okay. Alright, so this is showing you the same thing again. Okay. Here we go, plasmolyzed during hypertonic. You see that? So B is the representation of the plant cells, and then A is animal cells, and we talked about all of this right now. Okay. Alright, so we're going to talk about the facilitated diffusion. Okay, so it's as it sounds. It's facilitated, right? It's using, you know, protein, protein on its channel to facilitate that diffusion or transportation, right? So in a passive transport, do we use ATP? No, we don't. Okay, so we have basically, let's see here. We have two types. Do you remember that? We have the channel protein, we talked about it earlier, and we have the carrier protein. The channel is a channel, right? It's a pathway. It's going to allow certain solutes in and certain solutes out, right? The carrier protein is also the same way. It's going to allow certain things in and out. However, the carrier is going to carry, remember how I told you, it carries it and it changes the shape and then it spits it out of on the other side, okay? So, facilitated diffusion is a type of passive transport, okay? No energy is required. Okay, so we talked about how the channel proteins provide corridors or pathways. Okay, we talked about aquaporins, how aquaporins are actually a type of channel proteins specifically for water diffusion, right? We talked about also, I don't know if we did, actually the ion channels. It's as it sounds, right? They transport ions, okay? Those ion channels, we call them gated channels, ion gated channels. And you will see how, you know, they're specifically in the cells, in our cells, animal cells, we call them sodium channel, sodium potassium gated channels, okay? So there are gates, they open and close in response to stimulus, which could be, you know, like chemical or electrical, okay, and we will talk about this in a sec. So, for example, right, nerve cells. So whenever we're talking about nerve cells, you know, what comes to your mind? Electrical impulse, okay? So, you know, in the nerves, right, we have the electrical stimuli, and as a result, basically we have, you know, potassium and sodium channels being, you know, opening and closing at certain times, so that we have basically the ability to propel the electrical signal across, okay? Okay, so again, other gated channels, right, open in response to chemical stimulus instead. So you got the electrical stimulus and you got the chemical stimulus. So the chemical stimulus is basically it's a chemical and it's gonna bind and produce a response out of that protein channel. Okay. Okay, so we talked about how the carrier proteins function. Okay, they change their shape and then basically they're involved in facilitated diffusion. Okay, there's no energy that's required. Okay, hopefully this is making sense. So we went over this like 3 times so far. So let's see here. So facilitated diffusion is still passive, right? Because the solid moves down its concentration gradient. Okay, down its concentration gradient means you're going from a higher amount of solute or a molecule or whatever you want to another environment that's lower in uh, of that same type of molecule. That's why it's you're diffusing that into the empty space that have a lower concentration of it. Okay, sorry about that. Oh, where did we go? Okay, yeah. Okay, so however, we have also some transport proteins that move the solutes against their concentration gradient. So from a low um, concentration to a higher concentration, so the other way around. So when you have that, what do you think? What do you think you want? You want to use energy, is that right? You want to use ATP. So you need actually energy right now to pump things out the other way against the normal way. Okay, so this is where active transport comes in. It requires energy and it basically requires ATP hydrolysis. Okay, all proteins involved in active transport are carrier proteins. You see that? Okay, are they channel proteins? No, they're carrier proteins in active transport. In passive transport, we can have both. Okay. All right. So, okay. So again, active transport is allowing those cells to maintain concentration gradients that differ from their surroundings. Okay, as we mentioned. So an animal cell, that's an example, has much higher potassium. Okay, and a much lower sodium concentration compared to its surrounding. Okay, this is controlled by sodium-potassium pump. So whenever you hear the word pump, there's energy that's involved. Okay, because it's like a machine that's basically driving this pump, and for that machine, whenever you have a machine, you need energy, right, to let it work. Okay. So what's happening is the transport protein is energized by transfer of phosphate group from that ATP. Okay. So this is basically like the order of how things go. So you're looking at a sodium-potassium channel pump, okay, or carrier, I mean, okay. So usually the way this goes is the way this each channel work is you have 3 sodium ions that gets pumped out of the cell. At the same time, 2 potassium ions come inside the cell every time this happens. So let's follow along. So sodium ions bind, right, to this carrier protein, as you can see here. Okay, so you have basically on the exterior, right, you have, as a result, high concentration of sodium because of what's happening here, and on the inside you have a high concentration of potassium. Okay, so when they're bound, on the carrier, the pump needs energy in the form of ATP, as you can see here. And so it does that by when the phosphate of the ATP binds to that carrier, and when it binds, basically, this carrier basically pumps. It allows it to do its job. Okay, that's the splitting of that ATP here is allowing this to have energy. Okay, so when this happens, it changes shape, as you can see here. Okay, and it pumps those sodiums outside. Okay, and when it changes shape in this situation because it's a sodium potassium specific carrier or gated channel. Um, those potassiums are gonna come in and get basically attached here. Okay, and when this happens, when the two potassium, as I mentioned, that are trying to go inside the cell, they get attached here. That phosphate molecule gets released. Okay, that's just the way it works. It gets released, and as a result, that carrier opens up to the internal of the cell and releases those potassium ions inside the cell. Okay, and the whole thing, go again and again, same way. Okay, and you have a repetition here of the whole thing. Okay, so 3 sodium outside, 2 potassium inside the cell. You can watch the whole thing, how all of this happened on your own. Okay, so just so I can give you a little summary here. So in a passive transport, we have regular diffusion that we talked about, from a high concentration to a low concentration. Okay. We have also facilitated diffusion. Do you see that? Facilitated diffusion uses 2 types of protein channels. We have the protein channel, and then you have the protein carrier. See that? There's no energy required to do any of this here on the left side. On the right side, we have active transport, and that's a carrier, and this is what we just saw with potassium and sodium. Okay, and this requires ATP. You see that, to do the work. And remember, this is going against the concentration gradient. Alright, so we're going to talk about the membrane potential now. Membrane potential is talking about the voltage or electricity of the membrane. It's actually electrically charged. Okay? So membrane potential is the voltage across the membrane. Okay? Voltage is created by the differences in the distribution of positive and negative ions across the membrane. Right? Always remember, the inside of the cell on the cytoplasmic side is always negative. Okay, the outside is positive. So this favors passive transport of cations into and anions outside the cell. So what are cations? So if it's mostly negatively charged on the inside, what does it want to actually favor? Negative or positive? And wants to favor the opposite charge, which is positive. So these would be cations. Okay. What about the outside? Outside, we said they're positively charged, so they're gonna favor what? Anions, or the negatively charged ions outside the cell. Okay, so let's see here. So the 2 combined forces, right? It's called electrochemical gradient. So just think about this word, electrochemical. So you have electricity and you have chemical gradients. Both of them are driving the diffusion of ions across the membrane. Okay? So chemical force, as this sounds, it's a chemical. Okay? It's driving the ions concentration gradient. You have the electrical force. Okay, that's due to those membrane potential differences. You have negative on the inside and the positive on the outside. Okay, an ion diffuses down its electrochemical gradient. So an ion has to take into account both the electrical force and the chemical force gradient, and it moves down its electrochemical gradient. Alright, so let's see here. Electrogenic pump, basically, is a transport protein that generates voltage, gets a pump, okay, and it generates voltage across a membrane, storing energy that can be used for cellular work, okay. Let's see here. So I think this is looking at the plant cells. Yes, plant cells. So what's happening here is you have uh, differences between the plant and animal cells, right? In animal cells, those electrochemical pumps, if you will, they're called sodium-potassium pumps. Do you remember that? So these are the channels or the carriers allowing potassium in and sodium outside the cell. Okay. So in plants, basically, and fungi and bacteria, right? It is a proton pump, as you can see here. Okay, and that actively basically transports hydrogen ions, as you can see here, outside of the cell. You see that? So the cell is negatively charged on the inside, so the H positive is being driven outside of the cell. Okay? So there's also a coupled transportation by a membrane protein. So coupled transportation means it's taking into account two different molecules at the same time. They're working at the same time. So it really is when it occurs when active transport of a solute, right, is indirectly driving the transport of other substances. Okay. So what's happening here is you have a proton pump here, and you just saw this last slide. Okay, it's pumping all those hydrogens outside. Okay, so we're looking at a plant cell here. Okay, pumps those hydrogens outside. So when you have a higher concentration of H pluses on the outside, okay, that second co-transporter, start working. Okay. And so it uses those H plus concentration on the outside, along with the sucrose, which is a molecule that it wants to transport inside. It uses it together with the H plus to bring the sucrose or the sugar inside the cell. Okay. So we can say that the downhill diffusion of a solute is coupled to the uphill transport of a second substance against its own concentration gradient. Okay? Alright, so which one would be the uphill in these two? The proton, right? Proton is going against its concentration gradient because you have, an H plus here and you have, many H pluses here. So we're using ATP to drive them outside. Okay, so this would be the downhill diffusion, the co-transporter. Okay, so plant cells, as we mentioned, use proton pumps to generate an H plus gradient across the cell membrane. Okay, co-transporter couples the movement of H back down its concentration gradient to the active transport of sucrose into the cell, as you can see here. So they're being coupled together to go inside the cell. Okay, and this is how plant cells load sucrose or sugar into their veins for transport around the plant body. Okay, so animal cells use similar co-transporter, right? Um, to basically allow glucose to diffuse um, sodium ions into the cell lining of intestines. So here's what's happening. So the in order for the glucose to come into the cell basically uses those co-transporters as you saw with plant cells, right? Um, those co-transporters are basically potassium pumps. Okay, so basically whenever the sodium potassium pump is working and allowing things inside the cell, glucose come as well inside the cell at the same time. Okay, and this is actually therapeutic in uh, medically because with dehydrated like patients and people that have like diarrhea, and so they've lost a lot of water. The way they treat them is they give them basically either water or IV, right? With both um, solutes, okay, or salts, along with glucose, because they kind of work together as we saw previously, using those co-transporters to transport both at the same time glucose and the solutes inside the cell, and thus patients won't be dehydrated anymore. Okay, so let's see here. The bulk transport across plasma membrane occurs by exocytosis and endocytosis. We talked about this, right? Those are the larger molecules like amino acids, glucose, and so on, right? Let's see here. So these basically, come in and out with endocytosis and exocytosis using vesicles. Okay. Now the bulk transport, obviously, they're bulk transports, so they require energy. Okay, the cell's not gonna be like just do all of this, with no energy. Okay, you can watch the animation. So the exocytosis is, as it sounds, and we talked about this, right? The molecules that are being produced inside the cell, they exit via those vesicles, and this vesicle gets fused with the cell membrane, and basically, because of that, the the molecules is able to get out to the outside environment of the cell. Okay. So this would be like when cells in the pancreas secrete insulin by exocytosis. Okay. Now, an endocytosis is the other way around, right? Endocytosis is when the cell basically is uptaking, molecules into its vesicles. Okay. So we have basically three types of endocytosis. Okay. You have phagocytosis. I know they're very similar. They sound similar, but there are differences. Phagocytosis, we talked about it before. It's cell. It's when the cell is eating. Okay. Penocytosis and is when the cell is drinking. Okay. And you have a third type that's called receptor-mediated endocytosis. So let's talk about each one. So basically, this is the phagocytosis, and so the food or other particle, right, such as big molecules and so on, um, through endocytosis and specifically phagocytosis, uh, you know, a food vacuole is formed such as this, and it can be digested inside it. Okay. The penocytosis is when the cell basically um, try to take in um, droplets, okay, of the outside environment, basically filled with different solutes and molecules and so on. And so it basically creates this vesicle that's coated in a protein called PIT. Okay, and so as you can see, it transports things this way. Okay, and so you have the third kind, which is receptor mediated endocytosis, and that's the same what it sounds. It's got basically those receptors, and those receptors basically come in to the vesicle, as you can see here. Okay, and they get bound to the solutes get bound to different to specific receptors that they belong to, as you can see here. Okay. Okay, so we talked about all of this. Just read it. You know, we just explained the whole thing, right? What phagocytosis is, right? And you have basically the vacuole fuses with lysosome, as I mentioned, to digest this food particle. Okay. Now, penocytosis is, as I mentioned, those dissolved molecules dissolved in droplets are being taken up. Okay. Such as like drinking. That's why we call it drinking. The cell is drinking. So the vesicle becomes lined with those coat proteins. Okay. And basically, penocytosis is non-specific for the substances. It's not like this one. This one is specific, right? It's receptor-mediated endocytosis. So those receptors need to bind to specific solutes for that vesicle to be triggered to be formed. Okay. Okay. You guys can see all of that. Okay, and that should basically cover this chapter. I know it was a lot, and just study it little by little. Go over it slowly, and read the stuff in the book as well. And yeah, let me know if you have any questions, and see you in chapter 8. Have a good one.